signal that is transferred, the signal is perceived by a histidine kinase sensor, and then this is going to activate the response regulator, which will activate transcription of genes, resulting in a response. The transfer of this signal involves transfer of phosphate from this sensor to this response regulator, as we will see in a moment. So, excuse me a second. Acetylsyringon is sensed by the bacteria, and a sensor on the bacterial surface is going to be responding to the presence of acetylsyringon, which is a phenolic, and some sugars too that are produced. And the sensor is the product of the VRA gene in the VR regulon. So we saw that you have the VRA gene. So VRA codes for the sensor histidine kinase. Okay, so VRA codes for the sensor. And VRA localizes, obviously it's going to be perceiving something from the outside, it has to localize to the membrane. So VRA has, in a cartoon form, it's like this is the amino terminus, at the C terminus you have the histidine kinase domain. It has transmembrane domains, T transmembrane, that thread to the membrane twice. Here is once, sticks out and comes back with the histidine kinase in this on the cytoplasmic phase, and this is the outside. Okay, so if we look, then there are two domains to this protein. One is the sensing or the sensor domain sticking out, and the other one is the histidine kinase domain that is in the cytoplasmic phase of the cell. Okay, and I'll come back to that in a moment. Now let's look at VRG, which is the response regulator. And remember that VRG is that protein that I told you recognizes the Vir boxes and helps RNA polymerase transcribe the genes and operons in the Vir regulon. Okay, so VRG is this one here, and is the one that activates transcription of this gene or these operons. So VRG has two domains. One is receiver domain, and the other one is a DNA binding domain. Okay, so let's see how this works, and I'll show a diagram showing the grand picture this. So the signal, which is acetylsyringon, activates the histidine kinase of VRA. Okay, and this results in autophosphorylation of a specific histidine residue in the histidine kinase domain. And then the phosphate from this histidine, phosphorylated histidine, is then transferred to an ASP residue that resides on the response regulator, which is the VRG protein. Specifically, the ASP residue is in the receiver domain of VRG. So once this ASP residue is phosphorylated, so this phosphate from here is transferred directly here, it causes a conformational change, allowing VRG to recognize and be able to interact with the Vir box and activate transcription. Okay, so we have here perception of the signal by, let me move this out of the way here. You have perception of the signal by VRA. So here is acetylsyringon. You have VRA sitting on the membrane. The histidine kinase domain is inactive. Once you have this signal, it causes autophosphorylation of a particular residue, histidine residue. There are many, but it's only one that it undergoes phosphorylation. And now this phosphate from this histidine residue is transferred to the ASP on the VRG protein, causing a conformational change that activates or allows VRG to bind to the VR boxes and activate transcription of VR genes. Oops. So here it is. So VRA in the histidine kinase has a particular histidine that is phosphorylated. This phosphate is then transferred. So this part is the sensing domain is what senses the presence of acetylserine, which causes this autophosphorylation. Once this happens, then this phosphate is directly transferred to this ASP that resides on the receiver domain. Remember, there are many ASP residues, but it's only one particular one. Okay? And then this causes a conformational change, allowing the DNA binding domain to recognize the VR boxes. So, what do you predict? Let's say this particular histidine residue here, suppose this is histidine residue 100. What happens if I change that histidine residue to an alanine? What do you predict will happen? And the presence of acetylserine gone?
It wouldn't bind.
What do you mean it wouldn't bind? There's no binding of anything here. It's a phosphorylation. That is not binding. That causes, this is a kinase domain. It is autophosphorylation, means it phosphorylates. It's capable of using ATP to phosphorylate this residue. So what happens if I change that histidine residue to alanine? You need to think. Anybody? Anybody there?
Would it have to do with the properties of the amino acid?
Okay, well, so. Okay.
So I remember alanine is hydrophobic. So I don't think it would trigger the same reaction.
No, because it cannot. You have to have a specific histidine residue. Any other residue at this position is not going to work. Zero. No matter how much acetylserine you have, you are not going to have phosphorylation of alanine or leucine or threonine. Of the amino acid that can be phosphorylated would be like threonine and serine. You will not have that reaction. It will not happen. Therefore, if you do not phosphorylate, you cannot phosphorylate. Let's say you have an alanine residue, there will not be any phosphate to transfer here, and therefore you are not going to activate VRG. And you have to think, what happens if I mutate this to residue like phenylalanine? So you need to think, because I'll ask next time. And if not, I will ask it in a quiz or in an exam. Okay, so here is showing the VRA, which is actually is a dimer. So you have two polypeptides of VRA, which are sticking out of the membrane and involved. And here is the sensor domain that is involved in the perception of the sugars and the phenolics. Okay? And the kinase domain is going to be here in the C terminus. So activated VRG is able to bind the VR boxes, and it is the conformational change that allows VRG to bind DNA, the VR boxes. So phosphorylation of ASP is absolutely necessary for activation of the DNA binding domain of VRG. What you need to do when I post these slides and you look at this information is think, not just memorize, because that's what people tend to do, just memorize the diagram and this, what happens. No, you always have to think, what happens if you have a mutation here? This is genetics. What happens if you have a mutation? What happens if you cannot do that? Okay? So that's one way to study. So then, yes?
Sorry, this is Mike again. Can you go back to the diagram?
This one?
Oh, yeah, that one right there. So you were saying that if you mutated the aspartate to like alanine, you wouldn't be able to phosphorylate it because you said you can only phosphorylate histidine, threonine, and serine, right?
Well, no. I said some of the amino acids that can be phosphorylated are not all, but threonine, serine are commonly phosphorylated. In this particular case, you have to have a histidine. You cannot phosphorylate any other. You can have threonine, serine. It's not going to be phosphorylated. It has to be a histidine. This is why it's called a histidine kinase domain, which is the sensor.
Okay.
Here you have to have an ASP residue. You cannot have any other residue. You cannot have a histidine. You cannot have. That's the kind of thing you have to think about.
Okay, I understand that.
Yeah, you cannot change to any other residue. Whether it can be phosphorylated or not, that's relevant. It has to be an ASP.
Okay.
Okay, so I already explained what the two-component system was and how this is used in transfer of the information from the outside. In other words, how this signal acetylsyringon is perceived and how that leads to activation of VRG and then transcription of the VR regulon, activation by transcription. So as I said, two-component systems are present in fungi, plants, bacteria, but not in animals. And we know that from sequencing, since we have sequenced so many organisms nowadays that you can find the information readily. So how does the agrobacterium generate a tDNA strand? Remember that the tDNA is the only region of all this plasmid that is going to be transferred to the plant. And this is, the tDNA has this important information, which is how this bacterium is going to cause tumors, because it's going to be transferring this DNA, which has these plant growth regulators. And these plant growth regulators are going to be producing an excessive amount of auxin and cytokinin, and that's going to lead to tumor formation. It's going to disrupt the growth of the plant. Okay, so here I'm looking only at the tDNA. So the tDNA in these two triangles here means the border, so this TR, TL, or RB and LB. So there is going to be a nick on the bottom strand. So this is double-stranded DNA, top strand, bottom strand. Polarity, which most of you don't know what polarity is, 5' to 3' on this, 5' to 3' here. So it's going to be nicked on the bottom strand at TR, a particular sequence at TR. If you switch TL and TR, this is going to interfere with the process. It has to be TR. And the cut is done by VRD2, which forms a complex with VRD1. I just put D1, VRC1, and VRC2. So this is a complex of proteins. Okay, VRD2 is cutting or nicking here and binding covalently to the 5' end of this cut DNA. So it is analogous to what protein that we saw in conjugation. You guys are not keeping up with the material. So VRD2 cuts or nicks and binds covalently